Hi, everyone, and welcome to today's lecture on physical biochemistry. We're going to take a look at some energy conversion, water, and membranes. Okay, so before we get into some of the details of mechanisms, there are a few fun facts that I want to throw into today's lecture that I love when the textbook kind of gives you a little something that makes you think about things in, everyday life or things you may have encountered before, but really never thought about them in this particular way. So, one of the things that we see here is the idea that, water is critical to life. And you know that there are cold-blooded animals. Now, if they're not temperature regulating like the way that our bodies do, keeping everything inside nice and warm, how do they protect themselves from icy deaths in their body during the cold? And yeah, I phrase it that way because everyone calls me dramatic, and cold-blooded is also appropriate since everyone makes fun of how cold I am. But cold and sarcastic. But anyway, it's a good kind of point to think about, how are they protecting themselves? And let me just grab the laser pointer here. What's interesting is that they have antifreeze proteins. And what those do is they basically will go around and coat the surface of any ice crystals that start to form, and that blocks the interactions between the water molecules. So you see when the antifreeze proteins are coating the ice crystals, the water molecules can't interact with each other, and that will block the growth and recrystallization of ice that would end up fatal. Now, what I want you to do is circle star highlight the fact that what allows this to happen is that the antifreeze proteins are rich in threonine residues, the amino acid threonine. So circle star highlight threonine and make sure to have that in your notes that antifreeze proteins in these cold-blooded animals help to do this function because they're rich in threonine that interacts with water. And so that allows them to have that interaction with the ice crystals and with the water to prevent these icy, horrible deaths. Now, one of the terms we come across in biochemistry is bioenergetics. And what that is the energy conversion in biological systems. And a big example of this is photosynthesis, which we'll have a whole lecture and chapter dedicated to that. Now, there are three types of work that I want you to know that chemical energy can be converted into. Okay, the first one here is osmotic work, and that's basically going to maintain the varying solute concentrations across biological membranes. And so, osmotic, we're going to talk about in a later chapter, being the movement of water, and then you have it affecting concentrations of ions and various gradients and solutes. So, there's osmotic work, which again is across biological membranes. You have chemical work, which is going to be biosynthesis and degradation of organic molecules. And you can think of that as metabolism, right? The building up and breaking down of various things within the body. You also have mechanical work, which is muscle contraction. It's all about getting those gains. So we have osmotic work, chemical work, and mechanical work as the three types of work that we'll see in bioenergetics. Now, when I mentioned in bioenergetics, the idea of, building things up, breaking them down, metabolism, all of that, bioenergetics relies heavily on what's called redox reactions, which are critical to metabolism and critical to any kind of pathways because what redox reactions are is basically the transfer of electrons from one compound to another. It's usually sequential. You usually have, one giving electrons, one gaining electrons, and that's why you have it as redox, reduction and oxidation. And so we have over here in this little figure a listing of, what is it when you have electrons gained, oxygen removed, and hydrogen is gained? This side of the figure that represents Gur. So Leo says Gur, still one of my favorite memes, as simple as it is. Leo says Gur, so gain electrons is reduction. So this side, you can circle these three things. That's part of reduction, the red part of redox. And then these three over here, electrons are lost, oxygen is added, or hydrogen is lost. Any of these represent Leo, lose electrons, oxidation. So that's the ox at the end here. So this side, you can write above it oxidation. Now, within bioenergetics, what's very important is the study of thermodynamics. There are three types of systems when we look at thermodynamics, which is basically the transfer of things like heat, energy. In the three systems, okay, let me grab the laser. We first have the open system, which you see here and you see over here. In the open system, the example I'm going to give you is the human body. Okay, in an open system, you can see that matter, which here is shown as mass, and energy, which here is shown as heat, they are freely exchanged with the environment, okay, with the surroundings in an open system. So here you see it's uncovered in both of these examples. You have matter and energy going out and coming in. The next one is a closed system. And so now the example for this is now a covered beaker of water, or you can think of it as a covered pot on the stove. Okay, now that you have it covered, you are no longer allowing matter or mass to transfer between the outside environment. So now you only have energy, which shown here as heat. Okay, so you can have only energy exchanged with the surroundings in a closed system. That then brings us to the last one, which is shown here and over here. That can be seen as the examples of, a vacuum sealed item, or simply having a closed thermos with your coffee or tea in it, or ice water in it in the morning. So now with this system, you notice what's different about it from the others is now both matter and energy are being blocked from being transferred. So in an isolated system, neither matter nor energy is exchanged with the surroundings. Now, when we study thermodynamics, I want you to know the first and second law of thermodynamics. The first law, for that one right away, think of the term enthalpy. Okay, enthalpy, and you'll see it written on the chalkboard notes on the next slide. So now with this first law, you basically think of it as energy can neither be created nor destroyed. It only gets converted from one form to another. So, especially in terms of the forms that we were just talking about a moment ago when we looked at mechanical energy and, chemical. And we even mentioned, photosynthesis being a great bioenergetics example and conversion of energies. So now, when you look at this law, whenever you see delta H, I want you to remember that H represents enthalpy, and that is basically a term to represent heat energy. And when you look at this, I'm not going to make you memorize formulas or anything, but I want you to realize what it actually means when you see negative versus positive delta H, or again being enthalpy. So now, when we look at these formulas, just in case you're wondering, E represents energy, okay? So you have E final, energy final, the initial energy. Q minus W, Q is heat added, and W is work. Like I mentioned before, H represents enthalpy, E represents energy, and then you have pressure times volume over here. But like I mentioned a moment ago, I just want you to focus on this part here when we talk about these equations, and that is what does negative delta H tell you versus positive. So a negative tells you that the reaction that you're looking at is exothermic. It's going to release heat like you see here, whereas positive for enthalpy or delta H will tell you endothermic, and endo being within, that's going to absorb heat, as you can see here. Okay, now keep in mind this is the first law of thermodynamics, that energy can neither be created nor destroyed, only converted, and that you can look at it via enthalpy. That then brings us to the second law of thermodynamics, which is my favorite one because it has this amazing meme that can go along with it, and two, because it describes my life. So, a second law introduces the concept of entropy now. Entropy instead of enthalpy, entropy. And so, whenever you see delta S, remember that that represents entropy. And what entropy is a state of disorder. And so the second law of thermodynamics tells us that all spontaneous processes in the universe tend toward the side of disorder. So things like to go more toward disorder rather than going toward order. Okay, again, sounds like my life. But anyway, when you see this equation here, for instance, entropy of the universe equals entropy of the system plus the surroundings is greater than zero, that is telling you that, again, things in the universe tend toward disorder. Okay. And you can see this when we look at different states of matter. The more organized or ordered something is, such as a solid phase of matter, that's going to have the lowest entropy. Whereas when things are most disorganized, most disorder, bouncing around the molecules that you see here in the gas phase, that will have the highest entropy. Okay, so I could ask you which phase has higher or lower entropy. Okay, so now that we've looked at enthalpy, and you see it over here, H, and we've looked at entropy, S, that now brings us to the concept of Gibbs free energy, which is G. Okay, so this will take the first and the second law of thermodynamics and boom, give us Gibbs free energy. And it's basically showing us, or it basically means that we are looking at the difference between enthalpy and entropy of a system at a given temperature. Okay. And you can end up having, for instance, a delta G that is equal to zero. That will tell you that a reaction is at equilibrium, meaning the rate of formation of the products and the reactants is going to be equal. Or you could have delta G being less than zero or greater than zero. Okay. If delta G, the Gibbs free energy, is less than zero over here. And again, it's looking at reactants versus products. So that's why you're looking at this area here. The peak up here, that's activation energy. That's something else. So Gibbs free energy is over here between reactants and products. If it is less than zero, that tells you that you're looking at an exergonic reaction, okay, exergonic reaction up here, that's going to release energy. And as you can see here, it's basically favoring the forward reaction, meaning that the formation of products is getting favored over here. And exergonic, I want you to circle star highlight that exergonic reactions, meaning delta G that's negative, are going to be favorable and spontaneous, okay? Favorable and spontaneous, just like me. Whereas endergonic reactions, now you have a positive delta G. Those are going to be unfavorable, non spontaneous. They won't proceed unless you do something to help them push forward. Okay, and in this one, We usually see it occurring in reverse reactions because basically reactant formation is favored. So it's spontaneous in the reverse reaction, but not in the forward reaction. So again, if you see delta G negative, that's spontaneous and favorable because it's exergonic. If you see delta G positive, that is unfavorable, non-spontaneous. We call those endergonic reactions. That then brings us to the concept of the equilibrium constant, which we see right here as K subscript EQ, and you see it over here. And basically, this formula here is to show you how Gibbs free energy is related to the equilibrium constant, but I'm not going to make you memorize this. Instead, what I care about in this concept is being able to analyze a formula like this. So when we look at the equilibrium constant, and we see concentration of C and we see concentration of D up here and then A and B down here, that shows us C and D, those are products, okay? A and B are reactants. So if you see a K that is less than 1, right? Less than 1, for instance, 1/2 is less than 1. So that would tell you that the bottom here, that is going to be greater than the top. The bottom greater than the top. Well, what did we say A and B are? That's the reactants. So if K is less than 1, what's favored? The reactants, the formation of the reactants. Whereas if K is greater than 1, such as 2, well, think about 2 over 1, okay? C and D is now the greater numbers. Well, what's C and D? Those are the products. Okay, so if the equilibrium constant is greater than 1, then the products are favored. If it's equal to 1, well, then that will tell you that the formation of both of these is going to be the same, right? So neither reactant nor products are getting favored if these are both the same, which will give you a K equal to 1. At that moment, when the equilibrium constant is equal to 1, the delta G is actually equal to 0, okay? And what did we say before about when delta G is equal to 0? That tells you the reaction is at equilibrium, okay? It's at equilibrium. Now, a moment ago, I told you that endergonic reactions are unfavorable. They're not spontaneous, right? You know, who wants that in their life? You want a little spontaneity, okay? So if those reactions of endergonic reactions are unfavorable, well, then you might say to yourself, okay, then why do we even talk about them? They're never going to happen if they're not favorable, right? Well, what ends up happening to allow endergonic reactions to exist is that you can couple an endergonic reaction with a highly exergonic reaction because we said exergonic reactions are very favorable. They're spontaneous. You want to do them, which sounds so inappropriate, but maybe it'll help you remember. Exergonic are the ones you want to do, whereas endergonic are unfavorable, right? Now, in terms of delta G, we said endergonic is a positive. So when you see a positive delta G, you're going to think, oh, we don't want to do that reaction. That's endergonic, it's unfavorable. Well, what can we do? Couple it to a very favorable, very exergonic negative delta G reaction. And a big one that we use all the time is ATP, basically ATP hydrolysis because that has a nice favorable negative delta G. And what you notice is now, once you've coupled these two together, the positive and the negative delta G, now you have an overall delta G that's negative. So this overall coupled reaction will move forward. It's now favorable. It's now spontaneous. Okay, so I can ask you, what would you do in order to make an endergonic reaction favorable? All you have to do is couple it with exergonic reactions. And we're going to see this when we go through metabolism and we go through processes one by one, such as glycolysis and photosynthesis and the citrate cycle, all of those. You're going to see that there are a lot of endergonic reactions that end up happening because they are paired with a whole bunch of exergonic reactions to make the whole process negative delta G favorable exergonic. Okay, so keep that in mind in future lectures. Now, in addition to energy conversion, we said we'd take a look at water because water is critical for life processes. It's very significant to us. More than 70% of the mass of a cell is water. Okay, so the answer to that first question is more than 70% is water of cell mass. Now, an interesting concept when it comes to water is you will notice that when water freezes, the density, once it's become a solid ice compared to liquid, is actually less dense. Okay, so it becomes less dense when it becomes a solid when it's frozen. And that's very important because you notice when you have a cold drink of water with ice in it, the ice is floating. And that's very significant in the world because when you think about aquatic life systems and environments, you notice because ice is less dense than water and it freezes and floats on the top, that allows aquatic environments to freeze top down, meaning that all of the organisms living in there can stay surviving. Okay, they still have their aquatic liquid environment. If the ice was more dense and the aquatic environments froze bottom to top, all of the fish, all of the organisms living in there would die every winter, every time it ended up getting cold and freezing out. So this is a very significant concept that the density decreases when water freezes. The other concept that I have on here, another great meme that I found, the other concept that I have over here is that water is a solvent. What that means is that water can dissolve things. The things that it dissolves are solutes. And once you have the solute dissolved in the solvent, we call that a solution. So, one of the examples that I give for that is if you are making chocolate milk. Okay, if you take the powdered chocolate cocoa, chocolate powder, or chocolate syrup, that would be the solute. You then put it into the milk and shake it up. That milk is the solvent that's dissolving that cocoa powder. And then, boom, you have chocolate milk as the solution. And chocolate milk is always the solution because that is the right answer to happiness right there. You could also think of salt water as well. So, salt is a solute that dissolves in the solvent of water, and that makes the solution of salt water. Not as yummy as the chocolate milk, though. I apologize for anyone who is lactose intolerant. Another significant aspect of water is that it's polar. The polarity of water is very valuable to us and to life because basically, when you think of polarity, this gives a shape that's basically ideal for non covalent interactions. The hydrogen ends up being able to form hydrogen bonds, which is shared kind of bonding or attraction with, electronegative atoms like oxygen or nitrogen. And so you can see the water molecules being able to attract and interact with other molecules that have those electronegative atoms. And it could basically form four bonds that are short lived. And it can help also in terms of what's called proton hopping, where you can have these hydrogen bonded water molecules in a field pass those hydrogen ions from one water to the next. And you get this, temporary hydronium ion and all. But basically, what's cool about this process is that it can make these structures where when you have one individual hydrogen bond, it's actually kind of weak. But when you have a whole bunch together, that ends up being strong. And we're going to talk about in a moment how that's valuable to DNA, but you could even see that in terms of water surfaces and tension and all of that, and even, little bugs running across, which we'll also talk about van der Waals with that. But hydrogen bonding is pretty cool and pretty valuable. Now, the polarity of water is also valuable to the solvent abilities that we mentioned a moment ago, because like dissolves like, and so the water being polar allows it to readily interact with other polar molecules and not so much with some of the nonpolar ones. And it also allows it to basically dissolve things like, for instance, in this figure here, sodium chloride, because you have the water molecule having the strong positive side, the strong negative side, the sodium chloride crystals having the strong negative and positive side, and you get an interaction in which you have ionic bonds competing for each other, and it kind of pulls that sodium chloride apart. And that pulling apart is basically a layman's way of saying dissolving it. Now, as I mentioned a moment ago, hydrogen bonds being weak, having weak non-covalent interactions, are very valuable in genetics. They're very significant to DNA because, like I mentioned, hydrogen bonds individually are very weak, but put them together and they're very strong. So having hydrogen bonds hold the DNA double helix together is valuable because you want that double helix to be strong as a whole molecule, stay together when there's many hydrogen bonds. But you also need to unzip that DNA. Think about DNA replication and transcription. And so having hydrogen bonds be weak individually allows you to one by one unzip the rungs of that ladder of DNA and open it up for DNA replication and for transcription. Very valuable. Other types of weak non-covalent interactions that are very valuable to biomolecules include ionic interactions. And so ionic, think ions, that means that you have bonding and attraction between oppositely charged atoms, and it'll be based on, the electrostatic interactions. And ultimately, the strength of this kind of bond will depend on the distance between those ions and the environment between them. And when you have this occurring in proteins, it's called salt bridges. Okay, salt bridges in proteins are the ionic interactions. And then the last one over here is van der Waals interactions, which you see in the figures over here. That's basically when you have an attraction by this momentary spontaneous change in electron density of molecules that are electrically neutral. Okay, and it's valuable because it allows for interactions between nonpolar molecules. So if you look at atom 1 and atom 2, they're both neutral and normally wouldn't be interacting, but you have these spontaneous moments where those negatives kind of pool in one area, the positive pools in another, and now there's that electrostatic difference that kind of causes them to suddenly be attracted to each other. Okay, so that's very valuable with van der Waals, and one of my favorite examples of that you'll see in the next slides coming up. So, one of the cutest examples of van der Waals is the fact that geckos can stick to walls and they can run up and down these walled surfaces. And that's thanks to van der Waals forces. And I tell my students the trick to remembering what's allowing geckos to stick to the wall and run up walled surfaces is think of the trick van der Waals to walk up walls. Okay, van der Waals to walk up walls. Basically, the adorable little feet of these geckos, they have tiny, tiny hairs, millions of them, coating the bottom of their feet. And basically, they have, these tiny hairs that split many times to make them very fine tipped, and it gives the hairs a very large total surface area. And so they're able to form a lot of contacts with any surface, and those surface to hair contacts end up being very adhesive because of the van der Waals attractions that we mentioned a moment ago. These fluctuations in charge distributions between neighboring molecules, even when they don't have to be, polar, they could be neutral forces. And what's interesting is that basically one adorable little gecko foot can support the weight of an entire human. And so scientists are actually studying this concept and the structure of their adorable little feet. And look at that adorable little face. And the scientists are studying this to kind of help us develop new technology with walking robots and super adhesives, all thanks to van der Waals. And so, just to review some of the terminology that we just used and kind of recap the fact that weak non covalent interactions are very important to biomolecules and to our cells and bodies and to many organisms. We have this figure here that shows you things like electrostatic interactions, hydrogen bonds, van der Waals. For these, you could also think ionic interactions. Okay, and all of these are important because it gives versatility to biomolecules and anything that helps affect the structure and the folding of molecules ultimately affects the function. So we always have a saying in biology and in chemistry, structure defines function. So these weak non-covalent interactions, such as hydrogen bonds, van der Waals, electrostatic ionic interactions, they're all very valuable. And just to help you visualize that value, just wanted to show you some of how, for instance, the non-covalent interactions can lead to things like hydrophobic effects, which basically allows for folding of globular proteins. And so you can see how you have a long chain of amino acids and they end up interacting with each other and folding. And, a lot of times they'll fold very particularly based on residues like hydrophobic versus hydrophilic. Hydrophobic regions want to cluster away from water, and the hydrophilic ones will want to be on the outside. Now, getting back to the concept of water, there's a very important term and concept known as osmosis. We've mentioned it before and we'll mention it again throughout biochemistry. Basically, that's the movement or diffusion of water molecules from high water to low water. And if you want to think about it in terms of solute, that's the opposite. So when you have high water, you have low solute, and when you have low water, you have high solute. Okay. So you can basically, think of it in terms of solute or in terms of water movement. And when you are looking at osmosis, you're a lot of times asked, what would happen to a cell in hypotonic solution, for instance? Hypo being low, tonic being the salt solute. So hypotonic means low salt. And what would happen to a cell in low salt? Well, I could draw out the mechanism of osmosis, but you've seen that before in a lot of other classes. Instead, I just like to teach students a memory trick where when you think of hypotonic, think hippo, right? A hippo, big fat hippo. So cells will swell in hypotonic solution, they take in water. Whereas hypertonic, which is a very salty, high salt solution, think of eating like salty Chinese food, you get very dried out. And for hypertonic solutions, cells will shrivel up as they lose water. Now, we say cells in general, but sometimes there's terminology depending on the exact cell you're talking about. So, for instance, if you're talking about red blood cells and they are in hypertonic, so hypertonic is high salt solution. What happens in high salt? Shriveling, right? Loss of water. If that's happening specifically to red blood cells, we call it crenation or becoming crenated. So that's when they become shriveled in hypertonic solution because they've lost water. The same thing when we're talking about plant cells, you have specific terminology. In hypertonic solution, when a plant cell is the type of cell that gets shriveled up and loses water, we call that plasmolysis. Okay, plasmolysis. Now, the other term that you hear with when it comes to plant cells and water moving in or out is turgor. Turgor is simply the rigidity, how rigid that cell is, the pressure that they have from that rigidity. And so when you could either have high turgor pressure, which is a very swollen cell, has lots of water, that would be in hypotonic, right? Because it looks like a hippo. It got big and fat. Whereas low turgor is what you see with plasmolysis. You have lost water, it's shriveling up. Okay, so keep in mind the terminology for osmosis. When we talk about water, you also hear ionization of water. When you have water ionized, it's going to be ionized to a hydrogen ion, which is H plus, and a hydroxyl ion or a hydroxide ion, which is OH minus. And we sometimes see ionization constant to basically look at that and to show the relationship between the concentration of those hydrogen ions and hydroxyl ions. And when you see this equation and you see the two of those concentrations on the same side of the equation, that tells you that if one increases, the other must decrease, okay? And that gives you fluctuations to pH within biological systems, okay? So what does this tell you? It tells you that the ionization constant looks at the relationship between hydrogen ions, hydroxyl ions, and the fact that if you increase one, you decrease the other. That then brings us to the pH scale, which is something else that you've seen in a lot of your other biology and chemistry classes so far, so we're not going to go into too much detail here. pH, what it actually stands for, is the potential of hydrogen, and it's referring to hydrogen ion concentration and is basically looking at a measure of acidity. Okay, so if you have a low pH, you're going to have something acidic. And if you have a high pH, you're going to be looking at something basic, like a lot of people I know. Now, when we are looking at this comparison, we have the equation here. I'm not going to make you memorize that, don't worry. When we talk about pH, you also hear the term buffers. And I want you to know that a buffer is going to be a solution that resists small changes in pH. And that's very important in the body because you're not going to want things like blood, for instance, to suddenly change their pH dramatically. So having buffers in the body is very valuable since they allow for us to resist changes in pH. So, speaking of the blood and how you do not want it to suddenly change pH, you don't want your blood to suddenly become very acidic or very basic. That brings us to the bicarbonate buffer system. So circle star highlight bicarbonate buffer and make note that this is what maintains blood pH, okay? Maintains blood pH. It involves bicarbonate, which is over here, this HCO3 minus, and it involves carbonic acid, which is this one over here in the center. And you'll notice the red arrows show you what will be going on in terms of alkalosis. So if the blood is starting to become too basic, or the teal or bluish arrows will tell you what goes on to protect you during acidosis if things are becoming a little too acidic. So you'll notice during acidosis, the teal arrows, you're going to want to shift toward carbonic acid formation. You want to reduce the amount of hydrogen ions and increase the pH so that it's not so acidic. Whereas during alkalosis, the red arrows, if you're getting too basic, you want to make things more acidic. You want to switch toward increasing the hydrogen ions. And so instead, you'll move toward bicarbonate formation. Okay, so please make sure in your notes that you write down what you are going to shift toward in alkalosis and what you will shift toward in acidosis, which I have written on the chalkboard slides right here. That then brings us to the Henderson-Hasselbalch equation. Students get very scared when they see any equation like this. But again, I'm not going to go crazy in terms of the details of this. I just want to point out that when you see this equation that looks at the relationship between pH and pKa, which is pretty much how weak or strong an acid is, it's called the dissociation constant. When you look at this equation, I want you to note that A minus versus HA, if it has the hydrogen ion, right, that's looking at an acid concentration, whereas this one, the minus, that one is looking at base. So make sure to write up here, you're looking at concentration of base, whereas down here is concentration of the acid. So now I want you to take a look at this formula, and I pasted on one of the textbook questions here. So it's asking you to solve for pH, so we don't know this, but it will give you the pKa of the solution, and it will give you the concentration of sodium acetate and of the acetic acid. Okay, make note that this one says acid. So I want you to send me in the Remind app right now, pause your video, and send me a picture of this slide and send me how you would write out the formula to solve for the pH in this question. And you can say, I have no idea, I'm so confused on this slide, or you can just send me pH is equal to and plug in the values. I don't want you to solve anything. Don't send me one single decimal or anything. I want you to show me what you would plug into where on this formula, okay? Make sure to send that to me right now in the Remind app. If you're not part of one of the Remind app classes, then send me an email if you just happen to be watching this from another university or from a high school, or because you just have nothing better to do with your life and you love the sound of my voice. What I meant to say is I appreciate the views. Okay, the last thing I want to show you today is just a very brief intro to some cell membrane information. We're going to go into a lot more detail in the upcoming chapters, though. So, when we look at cell membranes, basically, I want to connect the idea of the cell membrane to something we already talked about today, which is entropy. Okay, and if you think of entropy, well, that's disorder, right? The higher the entropy, the more disorganized and disordered something is. So if you think about a cell, the inside of a cell is very highly organized structure, especially, our cells with all of those organelles very well organized. And so when you compare that to the outside of a cell, which is, constantly changing, very fluid, new things moving all around, uh, that's a lot of disorder. So in terms of entropy, that would tell you that inside of the cell would be very low entropy, very organized. Outside of the cell would be very high entropy, very disorganized. So now you can insult someone without them knowing it. If you go to someone's room or their house and it's really messy, you could be like, "Oh, dude, you really like entropy, don't you?" And, who you could say, "What beautiful entropy you have here." And they'll think you're giving them a compliment and it's like, "Nah, that is not a compliment." The next thing that I want to mention in terms of the cell membranes is the idea of phospholipids, very valuable kind of structures because they are what we call amphipathic. That's not pathetic, that says amphipathic. And what that means is that they have both a hydrophilic end and a hydrophobic end. Okay, so the hydrophilic polar portion and it has a hydrophobic nonpolar portion. Okay, very valuable to us. It allows for certain organizations to the cell membranes that get formed, and it allows for hydrophobic effects to occur. So, again, hydrophobic things like to avoid water, so that gives the effect of arranging in a certain way to, really get yourself away from water. We know some people in your lives that you want to get yourself away from, and so that's what hydrophobic effects do, in terms of avoiding water because it's not good for it. So the four possible complexes that you could end up having based on hydrophobic effects of phospholipids are the phospholipid monolayer. So it's a one layer of phospholipids here. That's kind of tricky and troublesome if you have a lot of, water around because you these hydrophobic ones don't want to be around water. So this works for if you have air on one side, water on the other. In our bodies, when you have cells, you're going to want a phospholipid bilayer. That way you can hide the hydrophobic portions inside the bilayer, and then the outside portions can be exposed to water. You can also have what's called the micelles. So that's C over here. The micelles arranged in a nice little ball, and you notice tight little center there. So you're not having anything in there, and it's a hydrophobic kind of region, doesn't want water there. That's different from the final arrangement, which are liposomes, which will have an aqueous center. So this is going to be a bilayer, but it's arranged in a ball-like structure. Okay, so the micelle matches the monolayer, the liposome matches the bilayer, and I want you to circle star highlight the liposome because the fact that it has that aqueous little center is going to be very valuable for us in the upcoming slide. So I said the liposome arrangement would be very valuable. If any of you took virology or microbiology, this may look familiar to you. It's the idea that you can have liposomes used to get drugs or medicine into cells. And it could be very targeted because the liposomes can have, nice targeted cell receptors on the outside that bind to the particular cells ligands on the outside that you want that liposome to fuse with. And you notice it basically fuses with the cell membranes of your target cell, and it drops off that drug directly into that particular cell. So it can be liposomes can be synthesized as a very targeted drug delivery system. And if you took microvirology, you've seen that viruses can do this too. So a virus can steal a membrane from your cells after it has brutally murdered them and likes to wear its skin. And yeah, viruses are jerks, as you saw in the pandemic. And the virus can then fuse with cell membranes to inject their genomics into their genetic material into your cells. And so what you're seeing here, though, is the drug delivery system use, but it may look familiar to you from other classes as well. So, the last thing I want to mention today is the fact that membranes are fluid, and I'm not talking about them being liquid. That would be really weird. So, membranes are fluid. That means that they change their shape within, the structure. They're not static, they're not stuck in one rigid position. And so that brings us to the concept of flipases. Flipases are simply ATP dependent membrane proteins that will catalyze this movement, this flipping of phospholipids from one spot in a membrane to another. And there are four possible ways that a phospholipid can be flipped or moved from where it's located in a membrane. There's the rotational movement, meaning it could just spin around. There is temperature dependent, what they call semicrystalline change. Okay, so that's the movement that you see here. And then you have flip-flop movement. Okay, so in flip-flop movement, you see the movement going from top to bottom. Okay, you also have lateral movement, so that's going from side to side. Okay, so four possible movements of the phospholipids thanks to flipases, which are the enzymes that are going to catalyze that movement. And so that brings us to the other review questions that I want you to send me in the Remind app. Again, if you're not in one of my classes, but you want to know whether or not you would successfully get these questions correct, if you're studying for, AP Bio or MCAT or DAT, just send me a message to my email address, which is listed on my YouTube profile. And so for these review questions, make sure to send them to me right away in the Remind app if you're in my class. That way, anytime, day or night, I can answer, see where you're going in terms of studying and how you're doing with the material. Please make sure to always send me a picture of the slide so that I know exactly what question you are answering. That is it for today. Thank you. Send any questions my way and have a wonderful day.